firing, so 2 tuning curves. And I sort of said, maybe it's 2 cells, but he said, well, they're always 180 degrees apart for a start, which seems odd. If it's just 2 cells, why would they always be 180 degrees? But also, if I close the door and just record in one compartment, now they have a single tuning curve. So these are cells that basically fire one direction in one compartment and the other direction in the other compartment. So this cell fires when the rat is facing with the cue card on its left, when the compartment is lemon, and with the cue card on its left when the compartment is vanilla. So always when the cue card is on the left. In other words, the cell is sensitive to the layout of the environment and the cue cards and so on, but not to the odor. And that means that when the rat steps through the doorway and encounters the rotated cues, then the cell flips its firing direction. In fact, we wanted to call these flip cells for a long time, but we decided that was too flippant. So we called them bidirectional instead. So the interesting thing about these cells, well, there are a lot of interesting things, but one of the kind of important things that we found immediately is that these cells co-occur with head direction cells. So these cells, the head direction cell plots are on the bottom and the bidirectional cell plots are on the top. And this is the 2 compartments, left and right. And you can see that the head direction cell has a consistent global direction, whereas the bidirectional cell has flipped from one side to the other, so that in this compartment on the right, the cells have aligned tuning curves, but on the left, they have disaligned tuning curves. So in other words, the cells are not doing the same thing. Now, we've never seen that happen before with directional cells, that cells do different things. Up to now, it's always been the case that head direction cells would always act together in concert, and that's a consequence of that attractor dynamics that I mentioned earlier. Now, we dug into this a little further. We found something even stranger, which is that when we looked at the cells that have 2 tuning curves, while it's true that some of them have only a single tuning curve in the single compartments, we actually found quite a few that had double tuning curves in the single compartment, like this one here. So even though this recording was only made in the lemon compartment, you can see that the cell has 2 opposing firing directions. The other thing is that one of these tuning curves is slightly more robust than the other. And it was very reliable finding that in the other compartment, when the rat stepped through to the other side, the large tuning curve would reverse direction. So there's still information about which compartment is which, but also there's an ambiguity about the firing of this cell. So we call these cells within compartment bidirectional cells because they have this bidirectionality within a compartment, whereas the other cells only show it if you compare between compartments. So there's 3 kinds of cells in the RSC. There's ordinary head direction cells that always have a single tuning curve. There's these between compartment bidirectional cells that look like they have 2 tuning curves, but if you look on a compartment by compartment basis, it's actually singular, and they're flipping. And then there's these within compartment bidirectional cells that have 2 tuning curves. And we did a lot of head scratching about the last kind of cell and what could be going on there. And we sort of decided that there is some type of interaction going on in retrospeidel cortex between cells that are dominated by the head direction signal and cells that are being dominated by the visual information about landmarks. So this is how we conceptualize it. So this is the granular retrospeidel cortex, which is the part that's more connected to the deeper regions of the brain, including hippocampus and the other head direction cell areas like anterior thalamus. So we imagine that granular retrospeidel cortex is predominantly getting information about head direction, and indeed we really only found head direction cells in this region. Visual cortex is feeding in information about landmarks. We're not quite sure what that looks like yet, but there are cells that are kind of somehow responsive to the environmental layout. And then we've got the dysgranular retrospeidel cortex, which has a whole mixture. So it's got cells that are being predominantly driven from the granular, cells that are being predominantly driven by the landmarks, and then we've got some cells that are being influenced by both. And we think that these are the within compartment cells. So there's 2 questions. How do these patterns arise and what might they be for? So to try and answer this, we did some modeling and also some hand-waving kind of speculation. So Hector Page, who was a computational postdoc in my lab, built a ring attractor network that tried to simulate some of the phenomenology that we've been seeing in granular and dysgranular retrospeidel cortex. So the basic idea is that the three types of cells differ depending on the inputs that they're getting. So head direction cells are predominantly getting inputs from other head direction cells. So when the rat faces north, the north head direction cells are driving that cell, and that cell will fire, and so it also becomes a north head direction cell. A between compartment bidirectional cell is getting its inputs predominantly from landmarks. So let's say it's a cell that's sensitive to the cue card, for example. So when the rat faces the cue card, that cell is strongly driven to fire. So in the lemon compartment, it fires when the rat faces north towards the cue card, and in the vanilla compartment, it fires when the rat faces south, also towards the cue card. So these are landmark-dominated cells. And then these within compartment cells, according to this model, are getting two types of inputs. They're getting the head direction cell inputs, and they're also getting the landmark inputs. So when the rat faces north on its first encounter with the environment, it's getting two sets of inputs. The north head direction cells and the landmark cells are all active. So this cell will be strongly driven to fire, and it'll produce a big, strong tuning curve. And there'll be some Hebbian plasticity so that these inputs get reinforced. Now, when the rat steps through the doorway and faces towards the landmark in the south compartment, the landmark inputs will still be active, but now the south head direction cells will be active because the rat is facing south. So the combination of landmark inputs and south head direction cells is going to strengthen these ones as well. And that means that the cell is now getting some south head direction cell inputs. And so when the rat faces south, it'll be driven to firing threshold as well. Now, it won't be as strong as when it faces the north because it doesn't have the backup from the from the landmark inputs, but it's enough to make it cross its firing threshold, and so it produces the smaller of those two tuning curves. So that's what we think these within compartment cells are. They're cells that are integrating landmarks and head direction inputs. Now, why would you have cells like this? Oh, this is the same. This is going through the same thing. I'll skip over that because I've just said all of that stuff. So we built this sort of model, and our kind of reasoning was that the reason that you would have this kind of wiring is that this provides a substrate for doing the kind of weighing up of cue reliability that I talked about at the start, where the brain is having to decide between these two inputs. If there's a conflict between what my self-motion cues are telling me and what my landmarks are telling me, is it that my self-motion cues developed an error, or is it that the landmarks have moved, like it was an unstable landmark or something like that? This is the architecture of the model. I won't go through it in too much detail, but basically it's a combination of cells that are getting information from the environment that are on their way to becoming bidirectional cells, the between compartment bidirectional cells. And then we have what Hector called conjunctive cells, which are getting head direction and environmental inputs, and they'll go on to become the within compartment bidirectional cells. So he built this ring attractor and then let the simulated rat walk around and experience some environments, and not to go into this in too much detail because it's a bit too detailed, but he successfully got bidirectional cells to form by this Hebbian plasticity. And some of those were within compartment and some of those were between compartment, depending on the initial wiring. So the Hebbian plasticity was able to develop this network. And then the question is, well, what can this network do? And the idea was that with a network like this, you can do this kind of cue reliability estimation. And we know that the head direction system has had to do this in a number of different domains. For example, we've known for a long time that the head direction system is predominantly driven by distal landmarks and not proximal landmarks. So a proximal landmark is something that you can walk around, like a piece of furniture in your room or something like that, whereas a distal landmark might be the hill that I can see when I look out of the window, or something far away, or even to some extent the window itself, but something that you can't walk around. So the idea is that as you're walking around, a proximal landmark will be very unstable relative to your head direction signal. Sometimes it will seem to be in the north and sometimes it will be in the south. And so it has a very inconsistent relationship to the head direction signal at any one moment. So Hector simulated this by having the imaginary rat walk around an environment that had proximal and distal landmarks with these cells that had this varying degrees of coupling between the landmark inputs and the head direction inputs. And he managed to get the cell to learn to distinguish the proximal and distal very quickly, based on the statistics of encountering the cues relative to head direction. I won't talk you through that because it takes a bit too much time. But just to say very briefly that the distal cues very quickly self-organized, the network self-organized itself based on the distal visual inputs, but not on the proximal visual inputs. And that was also true for bidirectional cells, which only developed tuning curves when there was this stable distal landmarks. So the second thing that this kind of cue reliability system might be for is to allow the learning of landmark stability. So this is a similar problem, except that now, instead of the subject moving around, the subject is stationary, but the landmark is moving around. And this is a problem that the brain also has to sort of solve quite a lot. So, you're in an environment and there are these large things around, and some of them are stable and some of them are not stable. So, for example, this London bus is not stable. And you want to learn to disconnect your head direction system from the unstable landmarks. So we have some experimental evidence that head direction cells are very good at this. So in this experiment that we did in my lab a few years ago, Bex Knight, my PhD student, exposed rats to this landmark, which was a light that moved between two locations. And it could move either a small distance, like 20 degrees or so, or a large distance, up to 180 degrees or so. And the amount that it moved back and forth increased day on day. And she found that at the beginning of training, the head direction cells, which are shown in this with the solid line over here, reasonably reliably followed the landmark, although not quite, interestingly, but so there was some conflict, but they were reasonably kind of faithful to it. And then when either the discrepancy got too large or the system learned, I'm not sure which it was because they were coupled, but then the cells stopped following the landmark. So they had acquired some plasticity related to the landmark stability. So this constant conflict between the self-motion cues and the landmark. So the system had finally stopped trusting the light, as it were. Now, there's some evidence from humans that retrospeidel cortex might be where this type of learning about landmark stability takes place. So Eleanor Maguire did a very interesting set of studies looking at people's ability to assess whether landmarks are stable or not, so landmarks being large objects. And she found that there's quite a surprising range of people's ability to do this. So some people are very good at knowing that a bus is not stable and a lighthouse is pretty stable type of thing. And some people are surprisingly poor at it. And she found that retrospeidel cortex becomes active when subjects are processing this information. And the amount of activity correlates with their performance on spatial learning tasks as well. So there's some evidence that retrospeidel cortex is interested in landmark stability. So we think that this system might be the place where weighted cue combination and re-weighted cue learning might be taking place, and that this could be a really useful model system for trying to understand the neural substrate for this idea, which has been, in psychophysics quite well kind of worked through for a long time. And so so we think that it would be really interesting to look at things like the membrane dynamics of the inputs that are coming in and how plasticity takes place, and indeed even how the basic perceptual decisions are made, how the integration between the landmarks and self-motion cues takes place. So yeah, we're hoping we can tell people on this as not just an experimental tool, but also a really useful modeling tool. So thank you for your attention. I'll stop there. I hope you're still with me. Are you still with me?
Yeah. Mm-hmm.
Yeah, great. Okay, fantastic. We got through it that time.
All right. Well, I will provide the applause for the audience that cannot otherwise reach you. Thank you very much. That was a really interesting talk.
Thank you. Sorry about the interruption in the middle.
Yeah, I mean, I actually did my graduate work working with rats and was deeply acquainted with the place cell system, the head direction system as well, the ring attractors. So I think it's a fascinating model and a fascinating system to work with. Interfaces very elegantly with the hippocampal place cell system as well.
Yeah, I think it's been underappreciated in the past, so I'm lobbying hard for it to get more appreciation.
Is it harder to record from than the hippocampus? I mean, the place cells are like sort of right there under the.
Slightly, but not massively. So we usually get people recording place cells in the lab to start with because they are really quite easy, but they progress pretty quickly to head direction cells. Yeah.
Okay. We've got a few questions here from the audience. Let me ask the first one, which is by Conrad, who says that people have probably seen enough of him today, so he's going to decline to ask you. All right, so this is a great question that I think is very, it comes to mind when you're thinking about how these systems operate. When there is a disagreement that is big between the previous belief and the visual landmarks, there could be either just one estimate or two estimates, right? Is there evidence that animals can carry two beliefs at the same time?
Not that we have seen in, well, I mean, you could say, so let me backtrack. So head direction cells always seem to have a single estimate. So they always seem to be in no doubt about what they think. They might be wrong, but they always have a unitary signal. But I suppose you could say that the within compartment bidirectional cells with their two tuning curves, in some senses, that might be a reflection of some ambiguity in a way. So according to their head direction cell inputs, they think they're facing one way, but according to the landmarks, they might also be facing the other way. So you could sort of say that some of these mixed signals may be a reflection of an ambiguity. And then the job of the retrospeidel cortex is to take these different things and weight them appropriately. So to decide, do I trust the certain one or do I trust the ambiguous one? And which of the ambiguous ones do I trust, and so on.
So kind of related to that, is there a time course over which if a rat is placed into an ambiguous situation, you sort of see the system resolving the conflict and sort of deciding on one alternative? Or
not that we have been able to see, but we haven't worked through and looked at the really fine scale timing yet. I mean, we have seen, or not we personally, but it has been seen, like Kara Jezik in the Moser lab did an experiment with place cells, introducing, in essence, a conflict. What they did was they rapidly changed the visual cues from one environment that had been learned about to another environment that had been learned about. And there was this moment lasting several seconds where the representation in the place cells actually flickered between the two. So there's some type of wrestling going on, and then the pattern would stabilize. We have tried to look for stuff like that with the head direction cells. We haven't had enough of them at one time to see anything like this flickering, even if it exists.
Yeah, I guess presumably the more you have simultaneously, the finer the time scale you can resolve, right?
That's right. Yeah.
Then you can sort of instantaneously estimate, whereas if you only have like one, then you need a lot more time.
That's right.
What is showing you.
Yeah. We have tried to look for things like, because you can only sample the direction of the tuning curve. So if the system had flipped to a different state, you wouldn't know about that different state if you've only got the one cell. But we did look to see whether there was a variation in the spike count of the tuning curves that reflected periods of time when the system had gone into a different state. And the outcome was sort of ambiguous, actually. It was hard to interpret. So yeah, that's an open question.
All right, another great question here. What's the time scale of the plasticity between landmark cells and head direction neurons? In other words, how long does it take to form the association?
I think the only data that I can think of offhand is Jeff Taube's experiments with cue cards some time back, where it took up to a few minutes for the pattern to stabilize. We don't have quite the same situation because when the rat is placed in the environment, so the signal establishes immediately, like the moment the rat goes in there, the head direction cells are firing. but we don't yet know whether they've learned about the landmarks. And then by the time the rat steps through the doorway and the cells rotate, the flip cells, or whatever we call them, are rotating, they're firing, that's reflective of plasticity, but we don't know what the timescale of that plasticity was, because by that time you're several minutes into the trial.
Yeah.
So yeah, we don't really know. I'm guessing it's quick.
But it's not overnight. I mean, it's within the time course of minutes.
Yes. Yeah. Yeah. So the question is it seconds or even milliseconds? Yeah.
Right. Okay. Next subpart to that question: for the within compartment bidirectional cells, is it perhaps because there's still symmetry of the compartment even though you present a cue card? So I guess, is it more about the sort of geographical layout?
Yes. So in fact, a PhD student in my lab right now, Ningyu Zhang, has been doing the same experiment with 4 compartments, and we see tetradirectional cells. So cells with 4 tuning curves, and some of those are within compartment tetradirectional cells. So within a compartment, they have these 4 tuning curves. So it's very clear that that pattern is a result of experience of the symmetry of the environment. So that's why we think it's, to some extent, it's a pathological situation. I mean, an animal would normally never encounter this type of situation where you had absolutely identical environment but opposing orientations. That's not very natural. So I think we're kind of producing an abnormal pattern which is exposing an underlying mechanism. So it's nothing intrinsic. There's nothing special about 180 degrees as far as the cell is concerned. That all came from the environment.
Well, and if that were true, then you might expect that, if a single place cell or direction cell has those multiple coexisting representations, and that would suggest the rat was basically completely confused as to which direction was which, and was sort of to some degree simultaneously entertaining all the alternatives.
Well, one part of its brain is simultaneously entertaining them all.
Yeah. Right.
But the head direction cells remain unitary throughout all of this, so it does get resolved. There is some kind of consensus decision comes out of it, and I don't know what a rat thinks, but my own subjective experience of processing of directions is I don't ever have the sense of having 2 simultaneous possible orientations. I may have a sense of uncertainty. I think I'm facing north, but I'm not sure. But I never have a sort of a double subjective sense. So I'm guessing it gets resolved before it gets to the sort of rest of the spatial system, for example. Yeah,
I had the same experience actually. It's really strange when you have that feeling suddenly, and you can feel your whole mindset shifting. Like, oh, that's and just all of a sudden, place. Yeah. Now you can imagine your head direction cells just going zoop, like
it feels like they're swiveling, inside your head.
Yeah,
it's really a funny.
It'd be interesting to know if there was if there is actually a swivel, like in the sense that you sort of, your sense of direction actually moves through angular dimensions versus like teleporting from one side to the other.
Well, that's the question that that I kind of touched on with the ring attractor and the activity moving around the network when you add a landmark. There's a question of, do you suppress activity in one part of the ring and it pops up somewhere else, or does it actually move right the way around? So you do really kind of rotate. And there's some evidence to support both of those, depending on the amount of discrepancy and so on. Okay. So I think it may depend on the attractor dynamics, which of those happens.
Right, like the strength.
Yeah.
All right, third question here. Are there neurons in the mouse, or the rat, I suppose, that respond to specific landmarks in the visual view? Because in the fly, apparently no such landmark neurons exist.
Not well. So I mean, there are in the brain there are neurons that respond to specific objects, for example. But so when you go down sort of into the kind of temporal lobes, you find visual cells become more and more kind of multimodal until they become very specific, almost like kind of grandmother cells in the inferotemporal cortex and primates anyway. So even in rats, we can see some object sensitivity in places like, for example, lateral entorhinal cortex. Mm-hmm. But in this area where we're talking about in retrospeidel cortex, right close to the primary visual area, we don't really know whether there's any sort of precise landmark information. In fact, we know very little about psychophysics of landmark processing by the head direction system. Is the signal coming in as a highly resolved set of objects that have been identified by some part of the brain and fed back, or is it something much more just statistical about the patterns of light and dark, and is it a much lower level type of thing, as you might expect just coming out of primary visual neurons? So we don't really know yet. There's a lot of work to be done to figure out the landmark side of all of this.
Yeah. A thought that occurred to me when you were going through the different kinds of cells is that I thought that this might also be consistent with several different coexisting head direction systems that exist at different spatial scales. So like one at the global scale, maybe one at a sort of more micro scale, and they both sort of coexist as separate populations, but distributed within the same cortical area. Is there a reason that that's not true?
Well, we wondered about that. So we thought about environments in sort of different degrees of locality, if you like, or modularity, like, or maybe a hierarchy. So there are some cells whose job is to encode direction over the entirety of space. Some cells that are responsible for a smaller segment of space. Some of them that are responsible for a very small segment, and so on and so on. So we thought about that, and that was one of the motivations for the 4-compartment experiment, because what we thought we might see are cells that are consistent in 2 of the 4 compartments and then reverse in the other 2 compartments. In other words, their job is to look after the direction for the 2-compartment subset of the 4-compartment space, if you like.
Right, yeah.
But we didn't see anything like that. So
Okay.
we just saw either head direction cells, which are sensitive to everything, or these very local cells, which are sensitive to the smallest of the compartments. So we don't think it's a hierarchical sort of layering of scales, but we haven't really tested that to destruction, so it's not impossible. We don't know much about hierarchical spatial encoding, actually, in the brain.
Right. Well, I guess, in the hippocampus, there's this idea that you get the sort of finer grain of representation at the dorsal end, and then it becomes progressively larger place fields. But I guess that's not really larger environments. It's just sort of more coarse-grained.
Yeah, that's right. Yeah.
Right. So the 4th question, I think what it's getting at, if I understand it correctly, is whether or not those different groups of neurons that you were stipulating sort of participate in one large global attractor network, or do they have their own individual attractor dynamics?
Yeah, that's a really good question.
Okay.
So the head direction cells, the classic head direction cells, there's kind of strong evidence for attractor dynamics there. Nobody's ever really kind of pulled the cells apart. These ones in the dysgranular region, it may be that there are multiple sub-attractor networks, but it may just also be that they're not connected. They don't have the same attractor dynamics. Mm-hmm. And I think we need to probably record large numbers of them, multiple levels of Q conflict to be able to see whether we can separate them out that way. But if I just had to put my money on it, I would say that it's probably not an attractor network in that region, that those cells are more independent, and their job is just to kind of harvest directional information from the environment and feed it to the head direction processing center.
Makes sense. All right. Well, I think we've just about run out of time for this session. Again, thank you very much. That was very educational and entertaining. And I was learning about the head direction system. It's so neat. Thank you again for sticking around to give one of these.
Yeah, sorry about the glitch earlier, but we got it. We got it figured.
Not your fault at all. Crowdcast has been in touch with us, and we've sort of explained the situation. They're very apologetic, and they're going to try to do better.
Okay.
Yeah.
Cool. It's a fantastic facility. I'm really thrilled that this has gone so well, and I'm hoping that we can do more of these. So
yeah, it's worked out pretty well. Yeah, we're hoping to push this forward as a template that other people can adopt.
Yeah, definitely. Yeah. Okay. Well,
right,
thank you,
everyone.
everybody. And take
So
care.
Kate has officially closed out the first day of Neuromatch 2020. And we will be seeing you again. Remember that there's an eLife session preceding the Neuromatch sessions tomorrow morning. The schedule is current. We moved one of the Zoom sessions. One of the Zoom sessions had some problem people trying to interrupt it, and we've shifted that one to a new time slot. It's now a Zoom Room 7. And so if you look in the schedule for tomorrow, you'll see where a Zoom 7 has been landed. And that is a replacement for the one that was canceled today. So once again, thank you very much, and we will see you folks tomorrow. Thanks again, Kate.
Thank you. Bye-bye.
Mm-hmm.